good morning. First of all, I would like to thank you for the organizing committee for the invitation, and especially to Rebecca Williams for the personal invitation. So in my speech, I'm going to talk about nanobiotechnological approaches to treat fungal infection. I'm from the Universidade Federal de Goiás, UFG. It's the center-east of Brazil. So my research group would try to discover or to develop new antifungal therapy using nanotechnology to prepare drug delivery systems to conventional drugs or to DNA vaccines. So why to prepare or to develop this new antifungal therapy? Mainly because of these three facts: by the increase of fungal infections, mainly because of the immunosuppressive patients under organ transplants, or by the patients committed by AIDS and cancer therapy, and also because of the life expectation of the population that we have decrease of the immunological system. And also because of the restricted number of antifungal agents. It's mainly on the polyenes and azoles. And so we need to discover new antifungal drugs. And why prepare new formulations for the conventional drugs? Because the development of new drugs involve high costs in research and development. So it's bad for poor countries such as Brazil. So why to use, our group decided to use nanobiotechnology. Why is nanobiotechnology? Nanobiotechnology involves biotechnology on the nanoscale. That nano derives from the Greek word dwarf. It means very small. So 1 nanometer, it's equal to 10 at minus 9 meters. So at this scale, you can found different molecules and proteins and viruses, and the nanoscale using nanobiotechnology, you works at this scale, around 100 nanometers to 300 nanometers. So nanobiotechnology is already used to identify and to study cell structures and physiology of the cells, on diagnostic and imaging, and to treat and monitoring disease, on radiotherapy and chemotherapy, and mainly to prepare drug delivery systems. Why? Mainly because of the small size of these nanoparticles, so we can target direct this on the body, and then to reduce the amount of the drug needed to treat the disease, and also to reduce the toxic side effects of this drug. Here I'm showing some examples of nanoparticles used to prepare controlled released systems. Liposomes, polymeric nanoparticles such as nanospheres or nanocapsules, dendrimers, or magnetic nanoparticles. Our group works mainly with polymeric nanoparticles, nanosphere or nanocapsules. So the first example I'm going to show is Nano-AMB, nanoparticle containing amphotericin B. So this is a schema of our nanoparticle that is prepared with polylactic-co-glycolic acid, PLGA, to prepare a polymeric blend, contains the drug amphotericin B. In this preparation, we put twice the amount of the drug needed to treat the fungal infection that I'm going to show you our experimental model. And also, we functionalized it with DMSA acid, this molecule. Why you use this? Because this, using in another kinds of nanoparticle, we show, and we don't know exactly why, it presents a tropism to the lungs. So, since our experimental models of fungal infection is Paracoccidioides mycosis that affects especially in the lungs, we decided to use this DMSA to functionalize and to direct the nanoparticles to the lungs to treat to kill the fungus inside the lungs. In our results, I'm going to show you the fungal burden recovery from lungs from animals infected and treated with different formulation. here, the conventional formulation for amphotericin B or our formulation for amphotericin B, they present the same results to treat the fungal infection. But our formulation presenting a better result because it's possible to put to give for the animals 1 dose and each 3 days, because in 1 dose we can put twice the amount of the drug needed to treat this fungal infection. And also, this our formulation prevented the genotoxicity effects caused by amphotericin B that can be seen here. And I don't like very much to show this picture because it's a sad picture, but it's unnecessary. When you compare the animals, the physical appearance of the animals, we can see here the non-infected, or the healthy animal, or the infected, this 3-1, and treated animals with amphotericin B or with our nano-formulation. We can see here our formulation did not cause serious side effects on the animals. When we analyze the biodistribution of our nanoparticles, we can see here, after 4 or 8 hours, a high amount of the nanoparticles of the drugs in the lungs that we expected to be. So, in this consideration of our nano-amphy, it allows a sustained and gradual drug release, presented antifungal efficacy and fewer undesirable effects, and favorable extended dose interval because we put twice the amount of the drug needed to treat the fungal infection. So, after this, we decided to use the same strategy to delivery of adjuvants such as DNA vaccines to treat the same fungal infection, Paracoccidioides mycosis. So we used DNA vaccine from heat shock protein. It's from a gene HSPS65 from Mycobacterium leprae, and it's used in experimental models against tuberculosis, leishmaniasis, arthritis, diabetes, and arteriosclerosis, and also it showed efficacy against fungal infection, Paracoccidioides mycosis. So we decided to use this DNA vaccine inside the liposomes for intranasal administration, or within polymeric nanoparticles for intramuscular administration, and used the same model of fungal infection, Paracoccidioides mycosis. So as we are trying to prepare a therapeutic vaccine, we used this nano vaccine to treat the fungal infection. So we infected the animals with the fungus Paracoccidioides brasiliensis, and after 30 days, when the infection or disease was established, we start to treat the animals with 2 doses. And after the second dose, we sacrificed the animals after 60 days, and which elicited protective immune response. For our nanoparticles, we used for liposome 25 micrograms of DNA vaccine or 30 micrograms vaccine for PLGA nanoparticles against 100 micrograms of naked DNA vaccine administered only in the plasmid. So it's 4 times less DNA vaccine using on these nano preparations. And our results show that the long fungal burden on the experimental groups, it's possible to see here both PLGA using 30 micrograms or liposome using 25 micrograms, both reduced significantly the fungal burden in the animals, compared with 100 micrograms of DNA vaccine free or in plasmid DNA. So we believe it happened because the macrophage or the dendritic cells capture these nanoparticles and then activate this immunological system, producing protective cytokines such as IL-12 or interferon gamma, and then resulting in a better protection to the mice. So concluding, nanobiotechnology is an important tool to be used to develop alternative therapies against fungal infection, and both liposome or PLGA nanoparticles dramatically reduced the amount of DNA HSP65 vaccine needed to treat PCM murine model. Liposome intranasal administration is a very attractive route to human use, and Nano-AMB was able to reduce fungal burden in mice lungs at the same levels than amphotericin B desoxycholate, with the advantage of allowing a reduction in the number of injections. I'd like to thank also to the involved researchers, especially to Dr. Maria Sueli Soares Felipe, the head of the group, and Dr. Ana Amélia Boca, that was first invited to present here. Thank you.